At the end of last time, by the way, I think that Monday will be our last get together in this form, as Tuesday classes end. So at the end of the last time, we were talking about air, and that that was related to the fact that peripheral antigens are expressed to a certain extent in the thymus. And if you, we also have seen that if you thymectomize mice at 3 days of age, you come down with autoimmunity. Some months later in multiple organs. So, and that autoimmunity is due to the fact that after day 3 in the thymus, there are natural Treg cells generated, which can inhibit that autoimmunity. And these Treg cells have 3 properties. One is that they express CD25 on their surface, which is the high affinity IL-2 receptor. Other T cells also express that when they're activated, but regulatory cells seem to constitute. They also make TGF-beta and IL-10. And to cut a long story short, or to put in my prejudice, Treg cells, when they make TGF-beta and IL-10, those 2 cytokines respectively help in the induction of IgA in humans and IgG4 antibody responses. Moreover, induced Treg cells, which are not generated in the thymus but in the periphery, particularly on prolonged immunization through mucosal surfaces, share with natural Treg cells generated in the thymus that they have CD25 on their surface and that they make TGF-beta and IL-10. So just as an introduction to immune class regulation in a sort of backwards way, if we get prolonged immunization through mucosal surfaces, the gut, the lung, through those mucosal surfaces, then we will get actually IgA produced and IgG4 in the long run. And induced Treg cells are very prevalent at mucosal surfaces. So what is interesting about this is that IgA doesn't activate effector functions, and IgG4, as I believe I explained before, doesn't activate any effector functions, but actually is labile. It splits apart and comes together again, so it's bivalent, but for 2 different antigens. Did I cover that a bit before?
Yeah.
Yeah, okay. Some more recent ideas on immune class regulation. But if we go back in history to look at the really big stories that people have been trying to understand about immune class regulation, we've seen in the past that different conditions of immunization are required to get delayed type hypersensitivity and to get antibody produced. So antibodies are a very vague term, given that we've got 7 classes and subclasses of antibody in humans, but very often people just did look at IgG antibody, of which there are 4 subclasses, so it wasn't very precise. And even in today's day and age, they often don't look very carefully at what subclass of antibody is produced, which is very important because they're induced under different conditions. But in the 1960s, people found conditions under which they could give animals antigen and make them unresponsive for antibody, for inducing antibody. And They also found conditions under which they could get T cells that would inhibit antibody responses. And the way I try to bring things together, which is certainly not generally done, partly because people don't know the old literature, is that there are 4 conditions from a bird's eye view under which you get inhibitory T cells for an antibody response, and where you get DTH exclusive of antibody produced. And I talked about this way back, but I said we'd come back to it later. So these 4 conditions are giving antigen which is subimmunogenic for an antibody response, giving antigen supraimmunogenic for an antibody response. So those, and you get DTH, so I call those low zone and high zone cell-mediated immune deviation. And you can give an antigen with few foreign sites. It will only induce delay type hypersensitivity, and it will induce inhibitory cells for an antibody response. So as an antigen with few foreign sites by itself does not induce antibody, the way you do this experiment is you take a typical such antigen. So one is polyglutamic acid tyrosine, a random copolymer of glutamic acid and tyrosine. And if you give it, you don't get any antibody response. In order to get an antibody response to GT, you couple it to an immunogenic carrier, which has many foreign sites. So now the GT becomes part of an entity which has many foreign sites. And so you can get antibody produced to GT. So you take immune serum, and it will precipitate with GT alone. So you've got antibody to GT. Very often, if you give that GT alone, before you challenge with GT BSA conjugate, you find out that you have ablated the response. And this is due to the generation of inhibitory T cells, suppressor T cells, which suppress the response to GT BSA. So are there any questions on what I've talked about so far? If not, then there's another type of maneuver which renders animals unresponsive for antibody. You give antigen in a way that would normally induce a good antibody response, and then you give, together with that antigen, you give an immunosuppressive drug like cyclophosphamide, which actually alkylates DNA and kills dividing cells. So actually, by ablating immunocompetence, you get suppressor cells for antibody induced, and when you actually look at it carefully, you switch the response from an antibody to a cell-mediated mode. So this would suggest that actually, in a sense, you need a stronger stimulus to get antibody than to get a cell-mediated response, because an immunosuppressive drug can switch an antibody to a cell-mediated response. Are there any questions on what I just talked about? All right, so as I think all of you know, when you clone, you can clone CD4 T cells, and if you clone them from an animal that is making a delay type hypersensitivity response, you get out Th1 cells that characteristically produce interferon gamma. And if you immunize so that you get just IgG1 antibody, for example, in mice, or IgE, you get out Th2 cells that typically make IL-4 and IL-10. And so I think one of the really major questions in immunology is, how does antigen interact differently with the cells of the immune system to generate Th1 and to generate Th2 cells? And this has been a subject of great discussion over the last 20, 30 years. In this case, I think that many of the suggestions, or the currently popular suggestions, I have problems with. So I'm going to give you one view, and one of the virtues, I think there are two virtues of the view I'm going to give you, but you have to understand it's my personal view, is that there's a lot of evidence for it, but it also brings a lot of concepts together. So what the view is, it's based on the idea that to get activation of CD4 cells, you require CD4 cells to interact in a way I described last time, mediated by a B cell. And if you don't get CD4 cooperation, antigen will inactivate a single CD4 cell. Are there any questions about the framework that I'm going to discuss things in the context of? So one thing, so the idea I'm going to describe to you is called the threshold idea. The idea is that you need different amounts of cooperation to generate Th1 and Th2 cells. You need robust cooperation to get the generation of Th2 cells, and you require tentative cooperation, less strong cooperation, to generate Th1 cells. And so if we think of a situation where we have robust cooperation, if you can put that in your mind, so that means that they're talking very well to each other, the T cells, that would give rise to Th2 cells and antibody. How would we change that robust cooperation to get weak cooperation or tentative cooperation, which would give us Th1 cells? Well, first of all, if we had an antigen at a dose where we got robust cooperation, since all the interactions are mediated by antigen, if we reduced the antigen amount, the interactions would be weakened, and if we reduced it sufficiently, we would get tentative interactions in Th1 cells. So that consideration explains why low doses of antigen generates Th1 cells and higher doses generates Th2 cells. Any questions on that? But we also know that if we have a foreign antigen, very often we make a cell-mediated response first, and then it associated with Th1 cells, and then we get the immune response evolving to get Th2 cells generated. But what actually happens when antigen impacts upon the immune system is that the first thing that happens, or one of the first things, is that antigen stimulates helper cells to divide. So when helper cells divide, so long as the antigen dose is maintained, you increase cooperation, and you can go from tentative to strong cooperation. And that explains why you go from Th1 cells to Th2 cells. And this is true for protein antigens, it's true in for HIV infecting people. It's true for mycobacteria infecting mice, and Leishmania parasites, which can cause the tropical disease of cutaneous leishmaniasis. Is true for a lot of different antigens, and seems a very general feature. So are there any questions on that?
Can you quickly summarize the cooperation again, like how it goes from Th1 to Th2 cells?
Sure. So when antigen, okay, so when you've got Th1 cells being generated, it means the TT cooperation mediated by B cells is weak or tentative. So when antigen impinges upon the immune system, the T helper cells divide, so they become much more numerous. And that means that they can interact with each other much better. And therefore, you go from tentative cooperation to robust cooperation, and therefore you go from the generation of Th1 cells to Th2 cells. Does that help?
Yeah, thanks.
Okay, so there are two other things that it explains. And that is that if you have an antigen with very few foreign sites, it means essentially that there are very few helper cells specific for it. And if there are very few helper cells specific for it, even if you've got an optimal amount of antigen, you can only get tentative cooperation. So we saw that, like antigen GT, glutamic acid tyrosine, is an antigen with few foreign sites, and it only generates delay type hypersensitivity and not antibody. But if you couple it to a carrier and make it more foreign, part of an entity more foreign, you can get Th2 cells generated. And lastly, if you have an antigen with many foreign sites and you give it at a high dose or medium dose where it gives you a nice antibody response, if you knock out dividing T cells, making them non-functional, by giving cyclophosphamide, as I just explained, then you switch the response from Th2 or antibody to DTH and Th1 cells. And that's been observed. So these immunosuppressive drugs which kill dividing cells, that means that when you give the antigen, many of the CD4 cells which are specific for the antigen, they're triggered to divide, but then they die because the DNA has been damaged by alkylation by the cyclophosphamide. So all these different conditions, which variables of immunization, which are known to affect the response, can be explained by this threshold mechanism. Are there any questions on that? It's quite a lot to assimilate. So it turns out that this particular threshold mechanism is not really discussed much in the literature, but there is a tremendous amount of number of papers, very impactful papers on what controls whether you get Th1 or Th2 cells generated. And these are really of three kinds. One is that not only do you require PAMPs to activate CD4 cells, but different PAMPs affect the co-stimulatory molecules differently, and they're different co-stimulatory molecules, and they thereby affect the class you get, whether you get Th1 or Th2 cells. So that's, we might call the PAMP hypothesis for what controls the response. And just as we're in this, have already just been discussing the variables of immunization that affect the kind of response you get, there are two things which are really difficult, or three things which are really difficult. One is that you often, it seems to be a very general rule that you go through a Th1 response and it often evolves to a Th2 mode against pathogens, but pathogens do not change their PAMPs as this occurs. So how can the PAMPs actually control whether it's Th1 or Th2? Moreover, to foreign vertebrate antigens, you get the same pattern of Th1 to Th2. And also, if you take something like cyclophosphamide and you kill CD4 cells, some of them, 90% of them, you'd only expect to affect the size of the response, not the actual nature of the response, if PAMPs really determined the response. So are there any questions on that? So another type of hypothesis actually, it looks like I haven't covered that in the notes, so I'm not going to go into it. I'm going to just talk about another, I'm only going to talk about two. Another hypothesis is that there are different types of APC, in particular, different types of dendritic cell. And these different dendritic cells, if you can separate them on the basis of their surface markers. And if you give antigen to one type of dendritic cell, you'll get Th1. And if you give it to another type of dendritic cell, you'll get Th2. This kind of hypothesis doesn't really explain why different antigens go to different dendritic cells, or why the nature of the dendritic cell changes that is impacted when you change the dose of antigen. And of course, it doesn't explain the kinetics, that you go from a Th1 to a Th2 response. So I find these actually, quite honestly, personally, I find them really implausible. So are there any questions on what I've covered? Because I'm going to go into something pretty different shortly. All right, so if there are no questions, we'll just try to recapitulate a little bit about autoimmunity. First of all, we've seen that group A streptococci, infection by group A streptococci can give rise to rheumatic heart disease associated with damage to heart tissue, because the group A streptococci have antigens that cross-react with heart tissue, and you find antibodies that are cross-reactive that bind both to group A streptococci and to heart tissue. And that, of course, is explained by the fact that you require BT collaboration to generate cross-reacting antibody. So any questions on that? So it turns out that less appreciated in the literature is that when you get infected with group A streptococci, you also generate T cells that react with both group A streptococci and with heart tissue. So the same kind of a mechanism seems to apply to the induction of autoreactive CD4 cells, and that is what you'd expect if you have CD4 cooperation in the activation of CD4 cells. Lastly, there is a mouse strain called the nod mouse, which gets autoimmune diabetes, and it is really a remarkable model for human autoimmune diabetes in many respects. And when you look at this mouse, Autoimmune diabetes is, of course, a disease, autoimmune disease against the beta islet cells of the pancreas. And if you look in very young, meaning 3-week-old NOD mice, you find out that they have T cells, autoreactive T cells to one antigen. And as they get older, the repertoire of T cells greatly expands. And this is called epitope spreading. So the T cells start recognizing more and more epitopes. And this is really actually the kind of thing that you would expect if you had CD4 cooperation being required for the activation of CD4 T cells. So that's one example of organ-specific autoimmunity. Are there any questions on that? So there's also syndromes where the autoimmunity is much more generalized, and lupus is the obvious example. There are antibodies made to many different self-antigens. And people have made a model of what that might be due to. And it'll help to understand this model if you go to figure 8 on page 213. Here we're using the antigen bridge model to describe certain situations. So here on the left-hand side is a T helper cell recognizing the antigen, the nominal antigen, and giving help to a B cell. Of course, we could translate that into an MHC restricted model of BT collaboration, but we just, for simplicity, we're doing it this way as an antigen bridge model. What the model system, which I'm going to describe in a moment, suggests is that there can be situations where there are T cells which are not specific for the nominal antigen, but for an antigen that is expressed on the B cell surface, and that the T helper cells give all these B cells signal to in this abnormal fashion. And that means that a B cell, which would normally receive only signal 1 and be inactivated, is now activated. So you get, as it were, a situation that would normally result in B cell inactivation, you get B cell activation. And just in case people think this model is really going to be very artificial that I talk about, there are viruses which infect B cells, like Epstein-Barr virus, and where you get T helper cells, which then can cause polyclonal stimulation by recognizing, presumably, on a B cell which is not specific for Epstein-Barr virus, but is infected by Epstein-Barr virus and presents antigens which are viral, derived from viral antigens, presents peptides derived from viral antigens, in which case you can get this kind of abnormal induction. Are there any questions on that? So the way people discovered models for this lupus, the symptoms being very, very similar to human lupus, was that they were studying graft versus host disease. So they put parental cells into A cross B F1s. And of course, what normally happens there is that the donor parental lymphocytes of type A recognize the B MHC antigens as foreign, and they launch a graft versus host disease, and the recipient animal rapidly dies. And this recipient rapidly dies due to the response, and particularly due to CD8 cells, which are A anti-B CD8 cells cytotoxic T cells that start attacking the host cells. Any questions on that? So what people found out is if there was no class 1 difference in A and B, but only class 2, you didn't get graft-versus-host disease, but you got T helper cells recognizing the B class 2 antigens, and you got the situation described in figure 8. That you had A and B being MHC compatible right across the whole MHC, but you only gave CD4 cells from the parental A donor. You didn't get graft-versus-host disease, but you got a prolonged disease in which you got a lot of autoantibodies produced, very similar to lupus. So that is one reason why this is an interesting model, because viral infection or various other situations may give rise to this kind of abnormal induction. Are there any questions on that? So lastly, we'll return back to IR1 genes to see how they fit into this picture when we're trying to pull things together. To start off with, we know that there are strains of guinea pigs, some of which respond to polyl-lysine, and others of which do not respond to polyl-lysine in the form of an antibody response. We've now learned that the responders have class 2 molecules which will bind a short decapeptide of lysine, and the non-responders do not have class 2 molecules which will bind decalysine, and so you cannot get helper cells in non-responders, but you can in responders. So in the non-responders, if you take a B cell, think of a B cell which is anti-polyl-lysine, and if you now immunize with a polyl-lysine coupled to BSA, the B cell will take in PLL-BSA, process the BSA, present peptides on class 2 molecule on its surface derived from BSA, and T helper cells can recognize the BSA class 2 complex and deliver signal to the helper signal. So that is a basic way that B cells, the basic way that IR genes function, by presenting peptides from the antigen. And if they cannot present peptides derived from the antigen, there'll be no T helper cells induced. Is that all right?
Yeah.
Okay, so what people then found was that if they took larger, more complicated antigens, but they took very limiting doses of them, so they were limiting in the sense that some strains responded and other strains of mice didn't. And then they did the genetics of seeing whether you could get a response to limiting doses. You found out the same story. It was governed by IR1 genes that code for class 2 molecules. It was also found that IR1 genes control whether you get helpers or suppressors, which is consistent with a threshold mechanism. And it's also found out that in some diseases where it's very important whether you make Th1 or Th2 cells, infectious diseases, that H2 genes are important in controlling susceptibility or resistance. Leprosy is one example. It's very interesting that in HIV, it looks like in HIV, a chronic stable cell-mediated response is protective. You've got cytotoxic T cells that can lyse virally infected cells. This is a kind of response that long-term sex workers who are elite controllers and control the virus have. And it turns out the most important contributory locus for resistance to HIV and therefore resulting in elite controllers are class 1 molecules. So if you have certain class 1 molecules, you're much more likely to be an elite controller. It's not a simple relationship. It's controlled by many genes, but certainly class 1 molecules are very important. So that is a sort of a very interesting situation where MHC molecules again control the kind of class of immunity you get and in an infectious disease, whether you're resistant or not. So are there any other questions? So I think on Monday we'll go along for as long as there are any questions. I'm just going to go a little bit through the last session on immunology and medicine. And the first area is immunity and infectious diseases. And as I presume that probably all of you know, all vaccines which are used in the West work because they ensure a rapid secondary antibody response upon natural infection. And it stands to reason that this form of vaccination is only effective against pathogens that can be contained by antibody. And clinical observation suggests that antibody is not associated with protection in some diseases and infectious diseases. And two of these are HIV and tuberculosis. So there are really two reasons why antibody may not be effective. One of them is explained, illustrated on Figure 1, page 216, which is referring to sleeping sickness caused by Trypanosoma congolense, which is a protozoan, and it's transmitted by bites from the tsetse fly. And if you look at what happens in, or what is represented in Figure 1, it shows that the parasite has got on its surface a variable surface glycoprotein. This variable surface glycoprotein covers the majority of the surface of the parasite, rather as pine trees do in a pine forest, very close together. And this variable surface glycoprotein occurs in different variants. And initially, it has variant A, and a good antibody response is made. And what happens is the parasite numbers decrease, as expressed by the dotted line. And then the parasite, or some of the parasites, remaining ones, switch to variant B, and then anti-B antibodies made, and variant B declines, and some of the variant B parasites switch to variant C and so forth. And so you get this chronic relapsing disease called sleeping sickness. And so this is an example of the parasite escaping by having a very sophisticated way of varying the its surface target antigens on its surface. So this occurs over quite some time. Another kind of escape is due to of the same kind is influenza virus, but influenza virus, if you're infected by influenza, you don't get new variants from that one infection, but over time, you get new recombinant viruses, actually, which are generated usually in birds, and then they infect people, and you get a new epidemic because the old immunity, the memory, is not effective against the new variant. Another example of variation, but now occurring again within an individual who's been infected, is HIV. It's very well known that HIV has ways of making many variants. So if you're infected with one particular parental viral particle, as it multiplies, you make variants, and some of those variants will not be neutralized by the antibody that neutralizes the parental variant, and so the immunity generated, the antibody generated, isn't really effective against all the virus, and therefore you get a lot of viral variants and you get progressive disease. So that is the reason primarily why people think, or some think at least, that those people who actually resist in the long term HIV need to have a stable cytotoxic Th1 response, and people multiply exposed making such a response, for example, sex workers in Nairobi, as discovered by Canadian scientists, they are resistant to developing AIDS, and they have stable cell-mediated immunity. And other examples of probable likelihood of stable cell-mediated immunity being important occur in leprosy and TB. So I'll just talk a little bit about a basic approach to try and ensure you make a cell-mediated response. So about 20 years ago, or a bit more, 25, someone decided that they wanted to really establish a mouse model of an infectious disease called cutaneous leishmaniasis, caused by a protozoan parasite that lives in macrophages, and where infection could result in subclinical infections but no symptoms, or it could result in frank disease. And in those people who had subclinical infections, they expressed stable cell-mediated immunity in the form of delay type hypersensitivity and produced no or very little antibody, whereas the people who got lesions which spread across the body, that happened when they got antibody and their expression of DTH declined. And so what this gentleman did is he took a lot of strains of mice and injected them all with 1 million parasites. And he found out that most strains of mice actually made a stable cell-mediated response, and they didn't get disease. But certain strains of mice made a very transient cell-mediated response with 1 million parasites, and they got progressive disease. So people have tried to look at this situation to take susceptible mice and see whether they could find a rational way of making them resistant. And as we've seen, dose of antigen is very important in the kind of response you get. So if you take the most susceptible strain, which is called bulb C, it's a white mouse, bulb C. If you take bulb C mice and instead of giving them 1 million parasites, you give them about 100 to 300, they then make a stable cell-mediated response. They no longer, the response does not develop to make any antibody. And if you give these mice such an infection of 300 parasites, and then you wait for 2 months, and you give them 1 million parasites, which in a naive mouse of this strain will result in antibody and progressive disease. You find out you've got low zone cell-mediated immune deviation. The mice make a massive cell-mediated response to the parasite. The response does not develop into a Th2 antibody mode, and so you've got resistance to this parasite. Now, it's sort of interesting that in the West we do not have any vaccination strategies in humans which are operational. For pathogens contained only by cell-mediated immunity. But in the Middle East, people are really don't like the consequences of cutaneous leishmaniasis because scars are left, and particularly scars on the face are considered to be very, very ugly and to diminish one's prospects in life. And so it turns out in the Middle East, this parasite is transmitted by a sandfly. And what people do is expose babies' bottoms to the infected sandflies, and they get a little lesion on the bottom, which is not socially detrimental, and they get resistance. So actually, it's a sort of folk mechanism of possibly doing low-dose vaccination, which is used in less developed countries, in inverted commas, but not in the West. And in the West, obviously, people are looking for vaccination strategies against such things as HIV and tuberculosis and other pathogens which are contained by cell-mediated immunity. Are there any questions on that? So I won't have very much to say on Monday. I'll just go through a little bit about allergies, organ transplantation, and cancer immunology, its applications of what we've learned. The emphasis is obviously on basic immunology and not on these applications, but the applications are getting very interesting because it really looks like basic studies are opening up avenues. So I think I'll call it a day there if there are no questions. All right.
Okay. Thank you.
You're welcome. Welcome.